Hello guys, and welcome to our video on the overview of the cell. And today we're going to be covering a few major themes. Those are cell basics, the nucleus and ribosomes, the endomembrane system, as well as mitochondria and chloroplasts. So let's get started with our first theme, cell basics. So every cell is surrounded by a plasma membrane, which protects it from its environment. Now, if you remember back, plasma membranes are composed of phospholipids, which means that they have phosphate groups bonded to lipids. As we know, phospholipids are hydrophilic at their heads, and at their tails, they are hydrophobic. And so what you'll see is a lipid bilayer is formed in plasma membrane. So you're going to have all these heads are facing the aqueous outside of the cell, so everything outside of a cell. And then you're going to have the tails, which are hydrophobic, which means that they don't like water. They're going to be clustered towards each other to prevent them from contacting water. So the plasma membrane's main function is to protect it from its environment. So another thing that all cells share is cytosol, which suspends organelles. So cytosol is the main component of the cytoplasm, and that's the main liquid or semi-liquid substance that is in the cytoplasm. Cytosol, that is. Then we have the fact that DNA is stored in the nucleus or nucleoid. In prokaryotes, it's going to be stored in a nucleoid and that is not going to be enclosed in a membrane. But whereas the nucleus has a membrane surrounding it, as do all organelles in a eukaryotic cell. So eukaryotic cells have nucleuses, and if they have nuclei, then that means that they're going to have membranes surrounding all their organelles. As we know, DNA is the most important, one of the most important macromolecules because it contains the instructions to life. So cells, again, as I've already said, cells which have nuclei are eukaryotic, and those that lack nuclei are considered prokaryotic. So in eukaryotic cells, all the organelles are going to be membrane bound, which means that they're going to have one or more membranes surrounding them. But this is not the case in prokaryotic cells, which lack nuclei, obviously. So plant and animal cells is an important distinction we have to make, but the reality is that they tend to be similar for the most part, but there are a few differences. For example, plant cells have chloroplasts which help them conduct photosynthesis, while animal cells obviously don't, because animals don't conduct photosynthesis. Now let's talk about our second theme, which covers the nucleus and ribosomes. Now these are grouped together for a really important reason. That's because these two organelles handle gene expression, the execution of the instructions in DNA. Now it's pretty obvious what role the nucleus provides, because the nucleus provides the DNA, which is organized into chromosomes, which is stored inside that nucleus, and it's going to synthesize mRNA, which stands for messenger RNA. And so this is synthesized through base pairing, which we'll cover later. And so the whole process of synthesizing this mRNA is known as transcription. So then it is translated into amino acids by the ribosome, which it itself is made of rRNA, which stands for ribosomal RNA, which you can tell why it's called that, because it composes a ribosome. So the ribosome eventually forms a polypeptide, and so this polypeptide then combines with other polypeptides, or it folds itself into the correct shape, which forms a functional protein, which then executes whatever instructions were provided to it by the DNA/mRNA. So the main flow of genetic information, as we just described, is known as the central dogma. So basically, DNA goes to RNA, which ends up as protein, which actually executes the instructions that were provided to it. So that is the central dogma of, I guess you could say, biology in general. So then we have the nucleus. So the nucleus is surrounded by the nuclear envelope, where you could probably guess where the name came from because it surrounds the nucleus. And so this is another double membrane composed of phospholipids, similar to the lipid bilayer. So the nuclear lamina is a structure, think kind of like how the skeleton provides structure to the whole body. Think like that just for the nucleus. So the nuclear lamina is kind of the skeleton of the nucleus, so it consists of small protein filaments which help hold the nucleus together in the correct shape. So there's a region of the nucleus known as the nucleolus, and this is where the rRNA, ribosomal RNA, is synthesized. So this is really important because this is the main component of these ribosomes, as we've mentioned. So a really important distinction I want to make is that ribosomes can be located either on the endoplasmic reticulum, which we'll cover later, or they can just float freely in the cytoplasm and synthesize mRNA there. So then we have the endomembrane system, which is our third major theme. So the endomembrane system consists of lots of organelles. So it consists of the nuclear envelope, as we've described earlier, the endoplasmic reticulum, the Golgi apparatus, lysosomes, the plasma membrane itself, and vesicles/vacuoles. All organelles in the endomembrane system are connected by direct contact or by vesicles, which are also part of the endomembrane system. So vesicles are just these pieces of membrane that surrounds one of these organelles. And so this membrane piece is transferred between organelles, and these membrane pieces, they can hold various molecules within them to transfer between these organelles. So we'll cover the endoplasmic reticulum. So it's basically a huge network of membranes consisting of various tubes and sacs. But unlike various other membranes, this has space between it, and so it consists of these various sacs, and we like to call these sacs cisternae. That is plural. Obviously, one sac would be a cisterna. So there are two types of ER, or endoplasmic reticulum. That's how we abbreviate endoplasmic reticulum. So you can have smooth endoplasmic reticulum, which has no ribosomes in it, and rough endoplasmic reticulum, which has ribosomes in it. Now this distinction is really important because it influences the function of these two types of endoplasmic reticulum. Because smooth endoplasmic reticulum has four main functions: detoxification, lipid synthesis, carbohydrate metabolism, and storage of calcium ions. And so with the calcium ions, we see that lots of muscle cells tend to have lots of smooth ER because, as you'll learn later, muscles contraction requires calcium ions, as do various other processes. So oftentimes the location of the cell and the function of the cell influences what types of organelles it has. So then the rough ER synthesizes special proteins called glycoproteins, distributes transport vesicles to the Golgi apparatus, and it also synthesizes the membranes of both the plasma membrane and various other membranes. So obviously this is influenced by the fact that it has ribosomes. That's why it's going to be synthesizing these glycoproteins, primarily because it has ribosomes in it, which can help it synthesize these glycoproteins. So that shows you how form fits function. So then we have our Golgi apparatus. So once a product comes from the rough ER, or the smooth ER in that case, then what happens is it goes to the Golgi apparatus, and the Golgi apparatus is kind of like a warehouse. It modifies these endoplasmic reticulum products. Think of the endoplasmic reticulum kind of like a factory, and then think of the Golgi apparatus kind of as a warehouse. So it's going to do minor modifications to these products. It also sends out vesicles primarily to the plasma membrane, because those products can then be pushed out of the cell if necessary, which is the case most of the time. And it also manufactures certain macromolecules. One example is pectin. So then we cover lysosomes. So lysosomes are important digestive components, and so their main job is to hydrolyze macromolecules. So there's a certain process called phagocytosis, and so what happens in phagocytosis is that a vacuole, which is a really important storage organelle, it basically combines with the lysosome. It fuses with the lysosome, and the lysosome is full of all these enzymes. It has a super acidic environment, so it's really good for destroying these huge macromolecules. So it breaks down these huge macromolecules. In some cases, it can also be used to destroy bacteria and damaged cells, because a lot of time damaged cells can do even more damage to cells around them if they're not destroyed, and so phagocytosis is often used to destroy damaged cells. So the lysosome, for that reason, is the most acidic organelle, because that is the environment that is most suited to destruction of various macromolecules. So we cover vacuoles next. So vacuoles have various storage functions, and of course it differs by the type of vacuole and what cell these vacuoles are found in. So we have three main types. So we have food vacuoles. So this is the main type used in phagocytosis. So this is going to combine with the lysosome, and the lysosome's enzymes are going to destroy the macromolecule inside. But in protists, we have a special type called contractile vacuoles. And so contractile vacuoles, they basically contract. And so the water is, this creates a tension on the water, and this water is being compressed. And so as this water is being compressed, it gets forced out of the cell. And so this is really important because if there's too much water inside the cell, then that can cause various problems. It can cause, like lysosomes to be damaged. It can cause various other problems because water concentration is really important. So we also have central vacuoles, and these store water and other organic molecules, and these are found in most cells. So central vacuole, there tends to be only one of those, and every cell tends to have a vacuole primarily for this purpose. I also want to point out that food vacuoles are usually formed when phagocytosis needs to happen, so you won't find food vacuoles around all the time. They're only going to form when the cell needs to conduct phagocytosis on certain macromolecules. So we cover our final theme of mitochondria and chloroplasts. So mitochondria and chloroplasts are not part of the endomembrane system, and the reason that they are not part of the endomembrane system is because they have two membranes surrounding them instead of one, as do all the other members of the endomembrane system. So we'll cover each of these one by one. So mitochondria is really important because it is the powerhouse of the cell, as you may know, and they're also the sites of cellular respiration. And so in cellular respiration, oxygen is used to synthesize ATP from various fuels, such as glucose and fats. And so mitochondria are only found in eukaryotic cells. Chloroplasts are the sites of photosynthesis, and so photosynthesis basically takes solar energy and converts it into chemical energy. These are found in plants and algae, and obviously since plants and algae are eukaryotes, chloroplasts are also found only in eukaryotes. So we have to cover something really important, and that is the endosymbiont theory. The endosymbiont theory provides an explanation for the origin of mitochondria and chloroplasts, and also confirms various observations that scientists have made over the years regarding mitochondria and chloroplasts. So basically this theory states that the long time ago, billions of years ago, the ancestor of eukaryotes, which probably was a prokaryote, absorbed an aerobic prokaryote, which means that prokaryote used oxygen. So this aerobic prokaryote is now what is now mitochondria or chloroplasts in cells. So initially this tended to be a symbiotic relationship. In other words, one of these two members of this relationship could just leave and they would still survive. However, if they stayed together, then that would benefit both of them. So in general, they tended to stay together. But as evolution passed by, these two eventually became indispensable to each other. And so that's why today, if you try to extract the mitochondria out of a cell and you basically let it live alone, it will die because it needs the rest of the cellular machinery to survive. However, this does explain why we see various components of cells in mitochondria. For example, we see ribosomes within mitochondria. This isn't really something you see except in the endoplasmic reticulum. And we also see that mitochondria and chloroplasts both have DNA, which is really interesting, unique, because most organelles don't have DNA in them other than the nucleus. And that's like the control center of the cell. So mitochondria have two membranes. So the outer membrane is really smooth. It doesn't have many folds in it. However, the inner membrane has various folds. That's why we call it convoluted. And so these folds are again called cristae. So the mitochondrial matrix is everything inside the inner membrane. So inside this inner membrane, all the major things inside the mitochondria are there. So enzymes, mitochondrial DNA, and ribosomes are all found inside the mitochondrial matrix. Chloroplasts, on the other hand, are surrounded by two membranes, but inside the chloroplast is another system of membranes and sacs. Inside the inner membrane, that is, there are thylakoids. So these are various membranes and sacs. And so these sacs are often stacked on top of each other. And so we like to call each of these stacks grana, or individually we call one stack granum. The stroma is the fluid within the inner membrane but outside the thylakoids. And so this is where the DNA, ribosomes, and enzymes are located. And so the chloroplast, as we mentioned, is really important for photosynthesis, and it has various roots. For example, there's a really important piece of evidence that supports the endosymbiont theory, and that is that cyanobacteria show great resemblance to chloroplasts, and so that's why scientists believe that they are the ancestors of modern-day chloroplasts. So that's another example that proves the endosymbiotic theory. So I hope you guys enjoyed our video on the overview of the cell, where we covered a few major topics: cell basics, nucleus and ribosomes, the endomembrane system, as well as mitochondria and chloroplasts. Subscribe and like.